some of my students and the work which we have done under institutional projects. I'll be presenting on understanding the mechanism of differential seed vigor status and field establishment in sweet, dent, flint, waxy, QPM and popcorn genotypes under suboptimum temperatures and its improvement through seed enhancement treatments. Maize is a very important crop and it is primarily used as feed, fodder and for commercial use. We have maize in which there are some categories which are termed as specialty maize, on which I will be emphasizing my paper. So first is dent corn. Most of you must be aware, but some of the audience will not be knowing about the different classifications about specialty maize. So just a brief about them. Dent corn, there is a presence of cornus horn endosperm at the size, and the tip of the seed is little indented. And the next is flint corn, which is a tough endosperm, and it is having a very hard outer layer. The third is popcorn, where it has a popping quality and endosperm is very tough or tight. Next is waxy corn. Usually in maize we have amylose as a storage carbohydrate. Here it is amylopectin, because of which it is sticky and soft, and it is very much popular for in industry and as a feed among the people. And next is sweet corn. The kernel has 18 to 22% Brix sugar, and because of sugary, shrunken, sweet enhancers genes in mutant form, the sweet accumulation is the sugar accumulation is there in place of starch. The next one is quality protein maize. Here the endosperm has increased content of tryptophan and lysine, because of which it is a quality enhanced maize. Now, before I begin my presentation, I would also like to emphasize that Indian Agriculture Research Institute has developed 4 quality protein maize and 1 sweet corn hybrids. So, these hybrids are cultivated by the farmers in winter season and spring-summer season and also monsoon season. And the seed production is primarily done in South India in winter season. So, the emergence of the crop or field performance or early emergence or early vigor is very much important for successful crop stand, both for farmers cultivation and seed production. So we also need to see, and it is a problem both in quality protein maize and sweet corn, the early emergence. So this work is also important in this context. So for undertaking this work, because specialty maize genotypes has variation in seed compositions, because of which there is difference in seed germination, vigor and field performance, especially under suboptimum conditions. So for this, we have taken genetic stocks of all the 6 types, specialty corns, and then we have studied the seed germination under field conditions, both under optimum and suboptimum conditions, and then validated with the lab studies, then assessment of seed composition, and then going for the structural and physiological basis of the vigor status. Now, these are the different specialty corn genotypes, the seed pictures, and I would like to emphasize that sugary, shrunken and double recessive. Sugary is the type where the sweet corn has sugary gene, shrunken where it is shrunken gene, and double recessive where sugary and shrunken both are in mutant form in the recessive state. So it is highly shriveled, but the sweet content is high. Now, the study I have divided into 3 parts. First, the seed germination and field emergence studies. So, seed germination studies were undertaken at 15 and 20 degrees centigrade, which is suboptimum for maize, and 25, which is optimum for maize. And then, what we have found that under 15 degree temperature, in the lab condition, none of the genotypes germinated, because it is a primarily tropical crop and 25 degrees centigrade is optimum for the crop. So 15 degrees centigrade is not suitable even for initiating the germination. Now when we come to next, that is 20 degree centigrade, at that time what we have found that the sweet corn and quality protein maize genotypes showed poor vigor low germination and very poor seedling growth, whereas the other types, they perform comparatively better among the lots. And among 25 degree, which is the optimum, the sweet corns comparatively were poor in performance, although they germinated, and the dent, flint, pop corns, and especially the waxy corn, they performed very well. So what we can conclude from this, that under low temperature, 20 degrees centigrade, this sweet corns and QPMs do not perform, or they have a very low vigor. Now, why it is so? So we took some germination study, and what we have seen that during the course of germination, around 16 hours there is bulging of seed, and around 24 hours you can see superficial rudimentary radicle, and at 32 hours radical growth is seen, and sprouting occurs after 36 hours. So in 42 hours you can see the radicle. So what we have found, that among all the genotypes, the first initiation or sprouting of genotype in all the specialty categories, around 36 hours there is sprouting, but only in sugary and double mutants it does not occur. In sugary it occurs after 48 hours of in germination, and in double mutant it is 72 hours, just because they do not have sufficient reserves in them to provide the energy to the germinating seedling. Now when we have taken field emergence studies in the field under suboptimum conditions in winter season, similar studies or similar results were also observed, where sweet corns, sugary, shrunken and double mutant types performed very poorly in addition to QPM2, and very low germination and seedling growth was observed, whereas in popcorn, flint and dent and waxy types, which were performing better in the lab condition, they also behaved similarly in the field. That means they had good seedling growth and early seedling vigor and good plant stand. And there were high correlations value with field emergence and lab germination. So what I conclude here, that what observations we have seen among the specialty maize categories for seed germination under lab condition, similar results have been found in field emergence. Now seed compositional studies. As we all know, there is difference in physical properties, seed, shape, size, compactness, then type of starch, then sugar, oil content in the seed. The sugary types have high sugar and others have high starch. There is difference in test weight length, where popcorn and sugary types are comparatively smaller. Then this is very important, the rupture force, that is the toughness of endosperm. This is very important for its storability, then incidence of stored grain pest and also vigor potential. What we have found, the sugary types, they have very low rupture force, that is toughness of the seed, and because of very highly shriveling nature, the seed coat is also damaged or not very intact. Now, structural, physiological and biochemical activities which control the vigor status in specialty maize. What we have found, these are X-ray radiograms, and what we have found, the sugary types and shrunken and double recessive types, they have very papery or see-through nature. That means they are transparent. That means reserves, it does not provide compactness, because of which the reserves are not synthesized for initiating the seed germination. Now coming to the internal structures which were observed by scanning electron microscopy, and it was found that the starch granules, their size, distribution and compactness, the dent popcorns, which is the toughest, in popcorn they are spherical in shape, very compactly placed, that is there is no intermittent space in between. Similar trend was seen in dent, flint and waxy, although their sizes and structures varied among the genotypes. These are good, having high vigor. And these are the genotypes which have poor vigor. So quality protein maize, shrunken, sugary and double recessive types, they have very low starch granules, and they are very sparsely paced, and that too the shape is not uniform. Now coming to the seed coat integrity, what we have found, that the sweet corn types, The water-soluble sugars in the seed leachate and electrical conductance is also very high. This is because the seed coat or membrane integrity is poor. Now, for any germination to initiate, we have to soak the seeds, and during germination, there is synthesis of hydrolyzing, respiratory, and breakdown enzymes. So, what we find that otherwise dehydrogenase activity is poor or low. That means early respiratory activities are low, or the enzyme activities are also lower in the poor genotypes which have lower vigors. And the ROS mechanism, if we see, SOD or superoxide dismutase, superoxide content and H2O2 ion content, here also we find that sweet corns have poor quantity of them. So in this I can conclude by saying that the sugary and double mutant types have very poor germination. Medium or optimum, not so optimum, shrunken, quality protein maize and flint types, and good are there in popcorn, waxy and dent types. This also we correlated with the storage potential. What we found, the sweet corn and QPM genotypes, they stored only up to 6 months because of their specialized storage reserves and poor membrane integrity and lesser toughness of the seed coat. Flint up to 9 months and popcorn, waxy, dent up to 12 months. That is above 80% germination is retained up to 1 year. Now the second part of the topic was how to enhance them or how to take care of these genotypes under suboptimum conditions. In the last 5 years, we have more than 5 years, previous years, we have screened lot of seed enhancement treatments like hydro, halopriming, physical treatments, treatment with botanicals, bioagents, earthworm derivatives in both specialty and normal maize. What significant finding we have found that these seed enhancement treatments improve field emergence, speed of emergence, early seedling vigor, vegetative growth. They also preponed yield, flowering and improved yield. And most important, why the performance was better was good seedling growth and good root growth. That means root in the length, surface area and volume. And then another thing was that the specialty maize were more responsive to the seed enhancement treatments, and they were more responsive under winter and spring summer season when the conditions are suboptimum as compared to the normal season, and low vigor lots were more responsive to this. And the treatments identified were hydropriming, followed by dry dressing with thiram, a fungicide, halopriming with KNO3, magnetic priming, and seed treatment with Trichoderma viridae. This is a paper in which we have also given that all the seedlings, they have good root growth after giving all the priming treatments as compared to the control treatment or control where no treatment was given. So finally, if we have to see that if the farmer has to do, which treatment he should select, it should be economical, it should be easily, could be done by the farmer and can be like easily done to the seed. So what we have found, seed hydration for 17 hours followed by dry dressing with thiram could be recommended for farm seed enhancement treatment for uniform field emergence. These treatments we have taken to the farmer's field under extension programs, and there we have seen very good results in field emergence, crop yield and improved physical plant vigor. And these are the yield under farmer field in the prime plots. And then we also have validated these studies again in our farm, IRI farms, where quality protein maize and sweet corns, although they have poor performance as compared to the better genotypes, but hydropriming and thiram perform better. And similarly, the root growth was also better in the prime treatments, seedlings raised from prime treatments. And why this is important? Because early seedling vigor is very important under suboptimum conditions, at least in first 1 month. So under all these conditions, vegetative growth is good, root growth is good, and that's why they are able to take nutrients or survive better among the suboptimum conditions. So I would like to conclude my presentation by giving 2 conclusions, that the differential seed vigor status of specialty maize could be attributed to their variation in seed physical properties and compositional status, which influences their physical performance in field and lab both. And seed enhancement treatments, particularly seed hydropriming, seed halopriming, were effective both under optimum and suboptimum conditions, but they were more effective under suboptimum conditions. Finally, I would also like to acknowledge my organization, my institute, and higher-ups in my institute, and Dr. Firoz Hussain, who has provided me all the genetic material for undertaking these studies, Dr. Anjali Anand, a plant physiology, who has assisted me, and my 2 students and the technical staff who has supported me in undertaking this study. Finally, I would like to thank all of you for giving me this opportunity to present my work here and giving your kind attention for listening to my presentation. Thank you very much.